Okay, everybody, here is the second part of the video lecture for Chapter 18 on speciation. This will be the last video lecture for the semester. All right, so remember in the first part, or as we wrapped up the first part, first half of this lecture on Chapter 18, we were talking about Ernst Mayr and his concept of founder effect speciation, or what's called peri-Patrick. Not para-Patrick, but peri-Patrick. Remember that. And remember, Meyer was arguing that evolutionary change through founder effect speciation could take place so rapidly that it might not be recorded in the fossil record. And this, he said, could help explain why transitional forms between species are often rare or non-existent in the fossil record. And remember, we said that Eldridge and Gould, when they developed their theory of punctuated equilibrium in the 1970s, sort of rolled Meyer's concept of founder effect speciation and its implications for the fossil record into their theory, right? Okay, so the question then is that if we have founder effect speciation, where you have, for example, on the island of New Guinea here, a small population that migrates to a small island off the mainland and evolves into a separate species relatively rapidly. The question here is, or the important point to understand or try and figure out here is that how could that population, that small migrant population leaving and setting up shop on a new island, how could that result in the evolution of reproductive barriers that would then keep these populations, the migrant population and the mainland population, separate enough or limit gene flow enough that this population eventually evolves into a new species? That's the question. How could this process cause the evolution of barriers to gene flow? Well, to understand this, we have to go back to the concept of the adaptive landscape that we saw in the previous chapter. The founder population here is going to undergo a shift from one adaptive peak to another that is driven by genetic drift. So genetic drift is the key here to understanding why this works, okay? Selection, as we've already seen, cannot take a population away from one adaptive peak down into an adaptive valley and up to the next peak, because that would require the population to become less fit as its gene pool moved down or changed in such a way that it moved down into that adaptive valley and became less fit. Selection can't possibly make a population less fit. It's an optimizing force that constantly makes populations more fit, or works to do so anyway. But we said that drift could, if its effect were strong enough, if the magnitude of drift were strong enough, it could take a population through a valley, an adaptive valley, so that selection could then take it up the adjacent slope to another peak. We talked about that in the previous chapter. Okay. Well, now we're going to look at a little more complicated version of these adaptive landscapes. We're going to look at 3D mesh figures here for adaptive landscapes, all right? So this is a little more confusing because it's three-dimensional, and so you have to sort of, in a way, kind of be able to manipulate this figure in your head, because you've got three different axes going here. You have a y-axis, which is always going to represent mean fitness, and a one x-axis and a secondary x-axis. And these two x-axes represent the frequency of two different alleles. All right. So remember that we're sort of operating on the idea here that hybrids, heterozygote intermediate hybrids between two different gene pools, are probably going to be less fit, okay, due to disruption of co-adaptive gene complexes or whatever, right? So we have one population that is very fit, so its gene pool has been selected for and molded by selection to be very fit. We have a second one over here, also very fit. These represent two adaptive peaks, all right? Okay, let's look at this first adaptive peak to see what's going on. So here we have a situation where if you pull this, if you come down, you've got a high frequency of allele A1. So A1's frequency declines this way towards 0, increases this way toward 1. This peak is more toward the high end of that scale. So this means that you're going to have individuals here who are A1, mostly, right? Because higher frequency of A1, two A1 alleles are abundant. They're more likely to come together than an A1, A2, let's say, right? Okay, now over here we have the same thing going on with B1. So B1 is low in frequency there, high in frequency there. Okay, so if we had B1 having an intermediate frequency of say 0.5, then you could have that combined with, so you have B1, B2 maybe as a heterozygote for B, but A1, A1, that would give you an adaptive peak, all right? A1, A1, B1, B2 is not a full-fledged heterozygote. It's heterozygote for gene B, but that's not necessarily a problem. We're talking about the interaction between gene A and B. All right, but what if we have gene A that's at intermediate frequency, 0.5, A1 is at 0.5? Well, if A1 is at 0.5 and B1 is at 0.5, that's when you get the highest frequency of A1, A2, B1, B2 heterozygotes. Remember, when you're looking at a gene with two alleles, you get the highest frequency of heterozygotes when the two alleles are in equal frequency, 0.5. If A1 is 0.5 here, what does the other allele A2 have to be? 0.5. That's where you get your highest frequency of A heterozygotes. B1, B2 both being 0.5 is where you get your highest frequency of B heterozygotes. So A1, A2 heterozygote, B1, B2, that gives you an adaptive valley. Those F1 hybrids are not fit. They have very low fitness, okay? That's an adaptive valley. All right, now, let's imagine we have frequency of A1 being low. That means A2 is high. But maybe we also have frequency of B1 being intermediate, okay? So we'd have A2, A2 homozygotes with B1, B2 heterozygotes. That would give you that peak right there, and they are fit, okay? So this is sort of how you have to be able to read these three-dimensional meshwork graphs here, showing us adaptive landscapes. Now, some argue that a population is not likely to move across an adaptive valley like that, say from here down and then up to another peak, because genetic drift probably would not be strong enough to oppose the really strong selection that would tend to want to force that population back up to that peak if drift pulled it down, right? I mean, drift would have to be really large, the effect of it really large to get all the way down and then just to the other side where selection could then take it back up to that second peak. So some argue that population is not likely to move all the way down into that valley and across it because drift would probably not be strong enough to oppose selection against the move down into that valley. They propose a sort of workaround to this problem, though. They suggest that shifts between peaks may actually represent shifts between different co-adapted gene complexes. All right? So, for example, what I mean by a co-adapted gene complex, you may have A1, A1, B1, B1, right? That might be a co-adapted gene complex. You could have A2, A2, B2, B2 as a co-adapted gene complex, but you might also have this as a co-adapted gene complex, right? A1, A1, B2, B2. So, shifts between peaks may represent shifts between different co-adapted gene combinations, and individuals homozygous for both alleles would have the highest fitness, while heterozygotes are less fit. So, you have individuals here, homozygous for both alleles, high fitness. Same thing there, and same thing here. Those individuals are homozygous for both alleles, right? These are different co-adapted gene complexes, all presumably being fit. Now, what you wouldn't have being fit, for example, is this, right? That is a heterozygote for both genes, probably not going to work out so well, probably have low fitness. Okay, so this brings us to a little more complicated figure here for an adaptive landscape, all right? So let's take a look at this. Okay, so the idea here is that the frequency of genotypes, say for example A1, A2, B1, B1, depends on frequencies of the alleles, right? So again, on our x-axis we have the frequency of our two alleles, A1 and B1, okay, with 0 being there. We have our mean fitness again on the y-axis. it's possible that the population could shift from one peak. So here we have one peak. Sorry, I get my pointer to work. There we have one peak, and there we have a second peak. All right, so we have two peaks here. It's possible that the population could shift from one peak to another along what's called an adaptive ridge. An adaptive ridge, okay, that avoids that sort of crater or valley in the middle. That adaptive ridge represents hybrid genotypes. So, for example, here you have genotypes that are homozygous for both A2, A2, B1, B1, right? Here you have maybe A1, A1, B1, B1. So again, these are homozygous for both genes. They're going to be most fit. All right, what do you have in the middle? Well, the middle, this is called an adaptive crater, by the way, an adaptive crater, all right, right there. In the middle, you have, at a frequency of A1 being, 0.5 and B1 being 0.5, that's again where you'd have the highest frequency of heterozygotes, A1, A2, B1, B2. That's the adaptive crater, okay? Now, let's take a look and see why it is that this is an adaptive peak. All right, draw a line down. We have a high frequency of A1. So we know that that's going to be A1 homozygotes, all right? Now, if you come over here and drop it down, you end up right there on the x-axis for B, and that would be B1 being high in frequency, right? So where, and I'm going to kind of mess up this figure here a little bit, but where this one meets and where that one meets, that's where you would get A1, A1, B1, B1 homozygotes. They have high fitness, okay? Now, let me change colors here if I can. All right, okay. Now, what about this second peak right here? Well, here we have B1 being high in frequency, okay? But we have A2, or I'm saying, A1 being low in frequency. Well, if A1 is low in frequency, what does A2 have to be? High in frequency. So you draw your line up there and your line up here, and where they meet, you get that genotype. So A2 is high here, B1 is high there, that's an A2, A2, B1, B1 homozygote. That's the other peak. And again, here we have our heterozygotes, so 0.5 for both alleles, for both genes. Frequency of heterozygotes is the highest, A1, A2, B1, B2. Those are poorly adapted. Excuse me, so our fitness is low. That's our adaptive crater. All right. So how does a population then, a founder group, let's say, that leaves this population and moves away, how does their genotype shift to a different peak that then represents potentially the evolution of a new reproductive barrier between these two gene pools? Maybe that's the mainland, this is the island. Well, how do we get that shift? Again, the population could shift from the first peak to the second peak along that adaptive ridge, and that adaptive ridge represents hybrid genotypes, all right? Hybrid genotypes. So do this right here. What kind of genotypes might you expect? Well, that might be an A1, A2, B1, B1 genotype. Why would that be? Well, if we have, let's say a genotype right here, okay, come down, that's going to give you a frequency of A1 about 0.5. That means your frequency of A2 is also about 0.5. So at that point, that's why you get the A1, A2. But where are we on the B-axis, okay? We're here at high frequency of B1. So most alleles are B1. So here you'd have B1, B1. So again, that's an intermediate genotype, but you do have one gene being a homozygote while the other's a heterozygote. But it's possible that those could be fit. They might have high fitness. And so they would be that adaptive sort of ridge, allowing the population to shift from one peak to another. Okay, and that's the idea. So these hybrid non-heterozygote genotypes are as fit as the homozygotes, so the mean population fitness moves along that adaptive ridge, and thereby avoids that adaptive crater represented by heterozygotes. Okay. All right. So it's hypothesized that could happen as a migrant population moves away from the mainland population, and sort of just as a result of what drift did and what genotypes you happen to have, what alleles you happen to have in terms of frequency in the migrant population, you could have the new population shift to this peak as a result of moving across that adaptive ridge. Okay. All right. So parapatric speciation, parapatric, let's shift gears now. That's peripatric. Let's talk about parapatric now. Remember, parapatric is, there we go. Parapatric is where you have, two populations whose ranges sort of butt up against each other. You have gene pool 1 there, gene pool 2 here, and they're quite different. You probably have maybe a cline or something like that that causes reproductive isolation along that border, but there's very little overlap, and that's referred to as parapatric, a parapatric arrangement, okay? And parapatric speciation then could occur if gene flow across that boundary is weaker than divergent selection between adjacent populations with different selection pressures. The idea here is that even if you do have some small amount of gene flow across, selection might be strongly working over here in one way and over here in a second way, sort of keeping these populations distinct. And hybrids between the populations that might form in that hybrid zone would be less fit in either population, and that would result in a hybrid zone and a decline in allele frequencies, and that might keep those populations, those gene pools, distinct. All right, so what kind of evidence do we have for that? Well, there's a lot of evidence for this, actually. Here's one good example. What you're seeing here is a transect of samples that were taken from an abandoned mine into adjacent pasture land. There's a fence sort of shown right, well, not really shown, but there's a fence right there. This is pasture, grazing pasture, and this is an abandoned mine property, and the soil here has heavy metal contamination, okay, I believe lead and zinc, if I'm not mistaken. And so it's possible that we have some contamination in this pasture grassland on the other side of that fence, because these lead and zinc ions and things can move. All right, so here we have our sample sites. Distances between the sites are in feet that you see right there. Okay, so this really isn't a very long distance overall. And what you're looking at here is reproductive isolation in a grass species across a cline of soil contamination, heavy metal contamination in the soil. And so the way this is being measured, or the effects of this is being measured on the biology of the plants, is through their time of flowering, so days from some reference date. What they did is they took plants that were found along this transect and put them in a common garden that was not contaminated with heavy metals. And they looked to see the time for stigma emergence and anther emergence. And what they found is that right at that boundary where the fence was, where you have high contamination to the left and maybe low contamination to the right, there seemed to be a decline, okay, a dramatic change in these the timing of reproductive events. And so these individual grasses over here, these are all the same species, by the way, but these grasses in this gene pool might be reproductively isolated from the grasses in this gene pool, because take a look, their reproductive events are out of sync with each other. So how, for example, could pollen from this side of the fence in the contaminated area, how could that pollinate flowers, or eggs rather, in individuals on the other side? Because they're out of sync in terms of their timing. So that right there might be a reproductive barrier, that cline. And so maybe you have two parapatric populations, all right? Okay, here we have some more data on that. And what you're seeing here is self-fertility percent. So remember, plants can self-pollinate, right? And so what you see is that there's a relatively dramatic change, again, at that fence line in terms of the self-fertility percent. So these individuals tend to self-fertilize, whereas those over here on the right-hand side in the pasture contaminated pasture tend not to. And so, again, this is just suggesting that they're reproducing in different ways. And if they're reproducing in different ways, asexual versus sexual, self-fertilizing versus outcrossing, then that is probably something that could keep those gene pools separate. That's a parapatric situation. If that goes on long enough, it's possible that the individuals of the species on this side and those of the same species on this side might evolve into separate species because there's a reproductive barrier there apparently. All right, now let's talk about sympatric speciation. You recall that I mentioned yesterday, or in the last video, it was yesterday my time, but the last video, that sympatric speciation is the most controversial between allopatric, parapatric, peripatric, and then sympatric. Sympatric is the most controversial. The concept of sympatric speciation involves reproductive isolation of a subpopulation within an initially randomly mating population where there are no geographic barriers to gene flow. So remember, allopatric, you have some sort of obvious physical barrier, like a river, for example. But in sympatric, you don't, okay? So the theoretical problem with this model of speciation is that intermediate genotypes could act as conduits of gene flow that would prevent speciation. So you got intermediates that are being formed, hybrids that are being formed, and so they're causing genes to flow back and forth between the two groups that are now genetically different from each other. And again, sympatric speciation is controversial. Ernst Mayr, in fact, argued that it couldn't theoretically even occur. All right, but we do have some models that suggest that it might occur. And these models typically invoke disruptive selection based on an adaptation of subpopulations to utilize different resources, okay? So let's say, for example, that we have genotype A1, and let's say that that genotype is adapted to use resource 1, whatever resource 1 might be. And then we have genotype A2, it's adapted to use resource 2, whatever that might be. Well, if we have crossing between them, you might get an A1, A2 heterozygote that couldn't use either resource 1 or 2, would be poorly adapted to use either of those resources, and therefore that heterozygote would suffer from heterozygote disadvantage. They'd be less fit. And that would tend to keep these two gene pools distinct from each other, right? Okay, selection then might favor individuals more likely to mate with their own type of homozygote and therefore less likely to produce heterozygote offspring. So females who are A1 might preferentially mate with males that are also A1 because a female A1 mating with a male A2 is going to produce offspring that are less fit. And selection would operate against that, wouldn't it? It would. So you might then have A2 females preferring A2 males, and that's a reproductive barrier. It's essentially mate choice, female mate choice, that's keeping those two gene complexes separate. That could cause them to evolve into separate species over time. But again, all these guys are living in the same population. There's no geographic barriers between them. They come into contact with each other all day long. They just don't mate with each other. And that's a reproductive barrier. It's the same thing as having a mountain chain between them for its effect on how they reproduce and the minimizing effect it has on gene flow. Okay, so that kind of model for how sympatric speciation might actually occur requires two things. It requires what's called assortative mating. Okay, assortative mating. That's where individuals preferentially mate with individuals who have a similar phenotype as they do, all right? And it requires what's called trait preference. Trait preference, where various genes control female preference and also male traits, all right? So the trait could be, for example, coloration or tail length, as we've seen, for example, or maybe it's a character that improves fitness related to resource use. That kind of character is called a magic locus. Here the female may select a male because a character he has tells her he can utilize a particular resource, all right? So again, this is the female cueing in on male cues as a way to determine which males are best for her to mate with in terms of producing the highest quality offspring, which, contain her genes, right? All right. So now, as the last part of this chapter, let's talk about the pace of speciation. So we spent this chapter talking about modes of speciation, allopatric, sympatric, parapatric, the possible genetic models behind how they might occur, how barriers to gene flow, reproductive barriers might occur. Now, as the last part of this chapter, we're going to talk about how fast speciation might occur. Okay, how long does it take, for example, for an ancestral species to split into two descendant species? Well, the answer is it varies, certainly. But let's talk about this. So we can actually quantify the rate of speciation in two separate ways. First of all, let me just draw a box here. We have what's called time for speciation. Okay, time for speciation, or TFS. That's an F, not an E, sorry. TFS. Cannot write. Time for speciation. We also have what's called the biological speciation interval. Interval. All right. BS. Oh, done it again. BSI. Okay. BSI. Time for speciation is defined as the length of time needed for evolution of nearly complete reproductive isolation once it begins. Biological speciation interval is the average time between speciation events, or the branch points on a phylogenetic tree. So let's take a look. All right. So here we have a phylogenetic tree. Okay, the branch points again represent one ancestral species ultimately splitting into two descendant species. So the biological species interval, BSI, includes the time for speciation and then sort of a waiting time over which reproductive isolation has evolved, but again before a new round of speciation begins. So here, this sort of gray stippled area represents the time for speciation, again, the length of time necessary for evolution of nearly complete reproductive isolation. So at the top of this right here, at the end of TFS, you have nearly complete reproductive isolation, but then it takes a while for those two reproductively isolated gene pools to actually evolve enough differences to be considered separate species, right? So this time here is TFS, the amount of time needed for complete reproductive isolation. This amount of time from here to here, or from here to here, is the biological speciation interval, the amount of time it requires for two new species to evolve. You have to have TFS first, then you can have the biological species interval as a result of speciation occurring, okay? All right. So how long is the biological speciation interval, or what we call the speciation rate? Well, estimates of it vary, obviously, depending on the group we're talking about. For example, one of the most rapid estimates for BSI, or speciation rate, is found in the cichlid fish of the East African Rift Valley lakes, Lake Malawi, Tanganyika. There's another one I can't remember the name of. Oh, Victoria. There are upwards of 1,000 species of cichlids in some of these lakes, and it's estimated that they all diverge from the same common ancestor in as little as 300,000 years. In some cases, even faster than that. And there are good reasons why we think that may have occurred, which I think your book actually talks about a little bit, but we won't go into. But that's a very rapid rate of speciation, or a very short BSI, in other words. Contrast that with bivalve mollusks. Some of them have taken upwards of 10 million years to speciate into their present diversity. Horses have taken about 3 million years to speciate into their present diversity. So it just varies, right? I mean, it varies quite a bit depending on the group we're talking about. Now, what are the consequences of speciation as the last part of this chapter? Well, if you understand evolution, and hopefully by now at the end of the course you have some understanding of how it works, and hopefully a better appreciation for it, If you understand evolution, perhaps the most profound sort of realization that comes from that is that speciation, or the end result of evolutionary processes, is the driving force behind biodiversity. Evolution is the reason that the earth is inhabited by so many species. So when you look around you and you see this amazing amount of biodiversity that we've hardly even begun to describe, honestly, it's very profound to realize that that's a result of past evolution, right? And evolution's still going on, so new species are still being produced all the time. And that's again one of the underlying important points about understanding evolution, is that you have an appreciation for biodiversity. So speciation then bridges the gap between microevolution, which is small-scale genetic changes from one generation to the next, and macroevolution, which is the evolution of taxa above the species level, the evolution of genera, families, orders, kingdoms, right? Domains as the largest group that we have over very long periods of time. So what kind of patterns do we see then through speciation, and do we see any sort of pattern or correlation between morphological evolution and speciation? Well, Gould and Eldridge suggested in their theory of punctuate equilibrium that speciation promotes morphological evolution, change in form. Most evolutionary changes, they said, in morphology are triggered by founder events in peri-patric speciation, like what Meyer was talking about. In short, Gould and Eldridge were saying that speciation is thought to accelerate morphological evolution, right? The more rapid speciation you have, the more rapid morphological evolution you have. That's what they were saying. And in fact, a number of studies have revealed a pattern where speciation events explain much of the morphological variation among certain species of animals. However, in 2002, just before his death, Stephen Jay Gould conceded that there's no actual reason to believe that speciation actually causes morphological evolution. It may be, he said, that morphological change is simply associated with speciation and not a direct result of it. Well, if that's the case, then why do we seem to have this pattern where speciation seems to explain morphological variation? Well, changes in morphology may be correlated with speciation because reproductive isolation between populations allows any morphological differences between them to persist because of a lack of gene flow. Geographically distinct local populations will eventually come into contact again, and if reproductive isolation has not yet evolved, morphological differences between them will be eliminated through gene flow. Now, if reproductive isolation has evolved, sequential speciation events will capture morphological change in a character. They kind of lock it in, so to speak. So it will appear that the amount of change is a result of speciation, when in reality the amount of change is simply a result of speciation capturing or locking in and preserving variation that was present anyway, variation that was not eliminated by interbreeding. And that's what this figure is showing you, all right? So let's look at panel A here of this figure. So here we have the dark arrow meaning a geographic barrier is in place, the light arrow means it breaks down or goes away, and the red bar means reproductive isolation is evolved. Well, here we have some sort of geographic barrier. We see that over time, some character diverges between two now separated populations. They start to look different, let's say, right? But if not enough time has gone by when that barrier goes away for complete reproductive isolation to have occurred, then gene flow between those separate groups, now joined together, will eliminate that variation, and they'll homogenize again. Like taking red paint and white paint, mixing it together, you get pink, right? So that's one possibility. However, if we have reproductive isolation that has evolved now as a result of that geographic barrier, like a river, for example, with our cavefish that I talked about, then even if that barrier does go away, the two populations remain genetically distinct. My suspicion is that if the Ohio River were to dry up tomorrow and cavefish on both the north and south sides of it could get back together, they'd probably be too different genetically to be able to reproduce with each other, and their gene pools would not mix. So they would remain separate biological species. All right, so here we have morphological divergence that's captured as a result of the evolution of reproductive isolation. But then we can have the same thing happen up here again. We can have another barrier evolve or come into play and splits the population. Reproductive isolation evolves again, and now we have the barrier go away, but now they're different species and they can't intermingle or interbreed, and you see we have this variation that's being captured by successive reproductive isolation events. So the pace of speciation here would seem to correlate with the amount of morphological variation. And that right there is exactly what Gould was talking about just before he passed away in 2002. Okay, that's it for Chapter 18. That's it for the course. So hope you guys have enjoyed this, and we'll stop there.